And it's a pleasure to have this opportunity to talk about some of the work we do on the in silico NAM site at the FDA's National Center for Toxicological Research. So my talk is mainly going to focus on the drug-induced liver injury, or DILI. And as everybody knows now, the DILI is a big issue for the drug development and as well as for the drug safety review, the FDA. And close to 50% of the drugs that either failed in the clinical trial or during the post-market surveillance are not predicted well by the current preclinical testing systems. So there is a lot of the efforts was going on trying to find out whether NAMs can fill the gaps and to provide the better predictions for DILI. So at the NCTR, we also have several efforts going on. For example, we are working with the Emulate and to evaluate their microphysiological systems for the DILI prediction as well as for other endpoints. We developed in vitro models as well as the toxicogenomics methodology as well. So today I'm mainly going to focus on how we develop AI models for the DILI prediction. Now, this is a part of the much larger efforts that's going on to provide a consultation and to the CDER to assess the drug safety using the AI models. And we have a lot of the models that's been developed. And some of the models already been used for decades, such as like OWL2 models and DILI score. And these models has been used to evaluate either IND submission or the NDA submissions. Because the time limit, and today I'm only going to talk about the two new models, and one's the DeepDILI, another one is the AnimalGAN. Now, DeepDILI is just a fancy version of the QSR models, and basically we develop a lot of the QSR models, and then we're using the deep learning architecture to integrate this model together to provide the consensus predictions. And it's a binary classification, and we call whether it's a positive or negative, and the model was trained on 753 drugs approved by FDA before 1997. And then we evaluated the model using the drugs approved by FDA after 1997. That was around 250, and we tried to provide a little bit better perspective in the real world applications. So the prediction accuracy is around 70%. This is an in-house model, so it is very easy to be implemented in the FDA environment. We already did that part. And also the models has been independently reproduced by the NIEHS. So as I already mentioned, the animal models are only around 50% accuracy for DILI predictions, but our model is 70%. So of course, at the first glance, our models perform better than the animal models. But this is an unfair comparison, and because there is a lot of the compounds and we used in the comparison already been including the DILI training set. So in the DeepDILI training set. So what we did is we focused on 3 sets, and we only focused on these drugs which was not used by a DeepDILI training. And for these 3 sets, we do find out and DeepDILI is outperformed of the animal results with a very large margin. Now you already know there is a lot of the in vitro NAMs has been developed for DILI predictions, and this is the paper we published recently and provide a comprehensive survey on what kind of the in vitro NAMs has been used for the DILI predictions. But most of the assays we see is better than animal studies. So the question is, how DeepDILI compared to these in vitro NAMs? And we focus on the 3 in spheroid models, and these are public data, and one was done by Inspero, another one's Genentech and AstraZeneca. They're testing a lot of the drugs, and some of the drugs already been included in the DeepDILI drugs, which is not included in the DeepDILI training. And you can see the results as a fairly comparison. it's fairly comparable between the DeepDILI and the spheroid models. And then one day I presented these results to our chief scientist, Dr. Steve Kolosky, and I say, hey, this is the results. And he said, well, if you put the DeepDILI and a spheroid model together, what is the results going to look like? And it is a very interesting suggestion. So we did, and we still look at these drugs which is not included in the DeepDILI training, and we combine the DeepDILI results with the spheroid model and the results. And we made several observations. And the first, we found if the two methods agree, and we will be able to improve prediction quite significantly, reach to about a 90% accuracy for those drugs which both models say either positive or either negative. Sometimes for a drug, two methods disagree, and one model says positive, another one says negative, or vice versa. And we found the largest gain to combine these two models actually for that group of the drugs which have a conflict results from two methods. And we will be able to improve around 50%. So all in all, if you put these two methods together, we will be able to improve the DILI prediction by 15%. So the next question we ask is, okay, the DeepDILI plus the spheroid model seems to improve the prediction quite a bit, and how does this compare with the microphysiological systems? Of course, the largest data set published in this area is from the Emulate. So we look at the Emulate data set, and we find the 9 drugs which was not used in the DeepDILI training process, but with the data available from the spheroid models. And this is the column that shows is the liver chip results and from the Emulate, and the next 3 columns basically is the combination and the results. 3 different weight combinations. And you can see and very much consistent and agree each other. And only one drug, and ambricetin, these drugs and the liver chip says is negative, and but our model show is positive. So we take a deep diving and look at these particular drugs, and based on the information from the FDA labeling document, and it turns out, and the FDA labeling document indicates, and it is very difficult to discern whether this drug is going to cause liver injury or not. So now we're going to shift the gear a little bit, talk about the virtual animal models called the AnimalGAN. Now the AnimalGAN is a framework and to develop the virtual animal models and using the generative AI methodologies. And in our case, we're using a very specific generative AI method called the generative adversarial networks, or called GANs. That's why we call it AnimalGAN. And we published our first models in the Nature Communication 2 years ago. And since then, we developed the second model and the version 2. And this model is really interesting because we are focusing on specifically 9 rat liver biomarkers. So you may call this model as a virtual liver. And of course, we're still using the TGGATE data set to develop these models. But these models allow us to determine whether individual liver biomarkers fall within the normal range or exceed the upper normal threshold defined by the FDA and OECD guidelines. So the idea behind these models is we are trying to use these models directly in the regulatory applications in accordance with the FDA and the OECD guidelines. And just show a couple of the results. So we apply these models on the 355 drugs and from the drug matrix. And so we look at the concordance between the AnimalGAN results versus the experimental results. So basically, and we look at the experimental results to see how the experimental data to call the normal and abnormal. And then we look at the AnimalGAN synthetic data, how they call normal and abnormal. And then you just generate a simple 2x2 tables to look at the concordance. So the concordance varies across the different liver biomarkers, and but in average it's between 40% and to 60%, and in agreement between the animal study results compared to the AnimalGAN synthetic results. So I know 100% is good, 0% is bad, but 60% of what that means is good or bad. So we need to have a benchmark to evaluate the prediction results. So fortunately, we have 70 compounds which was tested by both TG gates and the drug matrix, and so we can use these 17 compounds to establish the baseline to assess the cross lab and the variations. So if we look at only these 70 common compounds tested by TG gates and drug matrix, so this is the results it shows on the yellow bar. So if you compare to the yellow bar, the green bar actually is very compatible, and in some cases even better. So we also assessed the performance on 28 preclinical drug candidates was provided by AbbVie. These drug candidates are never been on the clinical trial. Actually, that's all done in the preclinical stage. So we have these results. We also, again, compare the experimental results against our synthetic results generated by AnimalGAN. So this is a green bar shows here. Again, the results is pretty very significantly across these individual biomarkers. And for the ALT/AST, and pretty high, but for other liver biomarkers, it's fairly low. So again, and we compare our results with the intra-study replicability. Basically, if you conduct the same experiment in the same lab, by the same people at the same times for many times, what is the intra-study replicability looks like for each individual liver biomarkers? And so this is where the yellow bar was shows, and you can see the green bar and the yellow bar is very compatible. So just a quick recap and I presented, and we compared the DeepDILI with the animal studies. We do found DeepDILI and perform very well and relatively to the animal studies. And if we combined DeepDILI with the in vitro NAMs, and which was further improved the performance. And lastly, we look at the AnimalGAN, and we feel much confidence now. We feel the AnimalGAN will be able to generate the synthetic data, which might be useful in accordance with the FDA and OECD guidelines. So just a quick shout out to our team, and we have a team of the members of the NCTR called the Air Force team and the AI Research Force team. They did all the works, and I also like to thank my collaborator from CEDAR, as well as our chief scientist, Dr. Steve Kozlowski, and I'm just finished here. Thank you.